Hi everyone, and welcome to VodCast number 14 for Honors Biology at Desert Ridge High School. I'm Mr. Galladay, and today we're going to start looking at different cell organelles. And in particular, the first organelles that we're going to look at are ones that are associated mainly with animal cells. Now, most of the organelles that we're going to see in this presentation are found in both plant and animal cells, with one exception, and I will point that out to you when we get to it. But before we do, actually this is a good point to update your table of contents, update the organization in your notebook, and you can pause this presentation, and then when you're ready to go, just hit run. Okay, to start off, I'm going to show you some photographs. Now, these are all from animal cells, and more specifically, these are all human cells. And the thing that I wanted I wanted you to see from these is that they're all different. There is a huge variety of different types of cells. So when we look at these diagrams of cells, and you'll be seeing lots of diagrams, and you've already seen some of a quote-unquote typical animal cell. Please keep in mind that there is no one type of generic animal cell. These are muscle cells. These are cells that make up our bones. These are liver cells. These you may recognize as neurons. This one, I frankly have forgotten what this particular picture is of, and this is a cross-section of some skin tissue showing hair follicles, as well as a number of different types of skin, muscle, and fat cells. So just keep in mind as we look at this generic animal cell that they are in fact all very different. Okay, as we go through these, it's a good idea to make a nice big drawing on the left-hand side of your notebook, and then on the right side, that will leave plenty of room for you to take notes about the with the functions and so on. Okay, so a nice easy picture way to start is a cell membrane. This is just, we usually draw this as a circular thing. The thing about cell membranes, and sometimes I know students get confused between the cell membrane and the cell wall, is when you think of a membrane, think of something that's very thin and flexible. Think of saran wrap. Think of something that is barely even there. The cell membrane is made of a very special type of a lipid, which, as you recall, is a fat or an oil. And this is a lipid that has a phosphorus or a phosphate atom attached to it, phosphate molecule rather attached to it. And they form in a double layer. So the fancy word for that is that this is a phospholipid bilayer. Okay, and so we're going to have quite a lot to say about this particular structure in about a week or so. And then there are also sort of placed throughout that phospholipid bilayer, there are also various specialized proteins that we call membrane proteins, and they serve a variety of functions, and we'll be looking at some of those in some detail as well next week. The overall function of the cell membrane is to control what comes in and what goes out of the cell, and the phospholipid bilayer combined with these specialized proteins are what allow that to happen. Okay, so our basic cell right now has a membrane, and if you think of a soap bubble, right? That is a membrane that makes up that soap bubble, and that soap bubble has an inside and an outside. And so the outside of most of our cells that make up our bodies is a watery environment, and inside that cell is also a watery environment. And it's made of something that we call cytoplasm. Now, cytoplasm, as I just said, is mostly water. It also contains a number of different types of salts, some proteins, some nucleotides, some amino acids, polysaccharides, and so forth. One of the main jobs that all cells do is make proteins. So if you're going to make proteins, it's important that you have the building blocks that proteins are made of, and those, of course, are amino acids. One of the other things that most cells do is they make copies of nucleotide or of nucleic acids. And so to do that, you have to have some nucleotides around. So that's the main reason that those are there, and we'll see that in some coming weeks as well. Another important thing to understand about the cytoplasm is that there is a ratio of water to this stuff that's dissolved in it. And the ratio of that water in the cell needs to be fairly constant. And it's affected by the ratio of water outside the cell. Another way to think of this is if it dries up outside the cell, conditions get dry, then water is going to leave the cell. But we need to have a certain amount of water in there, and the cell, by spending energy, can maintain that correct water balance. And again, next week we'll be seeing some details about how that works. Okay, so our cytoplasm is a watery substance. The next thing that we're going to talk about is the nucleus. And important to remember that the nucleus is not just one thing. The nucleus I sort of think of as being a bag within a small bag within a larger bag. The larger bag, of course, is the nuclear membrane, the cell membrane or the plasma membrane. And then the inner membrane is the nuclear membrane. Okay, so you have this inner, sort of like a soap bubble inside of a larger soap bubble. And inside that nuclear membrane is where the genetic material, which of course is DNA and RNA, is generally found. So the main parts of the nucleus is that there is a nuclear membrane, also sometimes called a nuclear envelope. That nuclear envelope has some holes in it, which we call pores, and then it also contains that genetic material, contains the DNA, contains the RNA. The function of that nucleus is basically to control the activities of the cell. The nucleus itself doesn't do that. It turns out that that is actually done by the genetic material, the instructions that are encoded on that DNA and RNA, and are executed by ribosomes. Okay, so as I just said, that genetic material really acts as a set of instructions that are carried out by machinery outside of the nucleus. Okay. So the next thing to talk about are the machines that carry out those instructions, and the machines that are found outside of the nucleus in the cytoplasm are these things that we call ribosomes. The best way to think about ribosomes, I think, is to think of them as little teeny tiny protein making machines. And then the instructions for making those proteins comes from the DNA and RNA that is inside the nucleus. In about a month, we'll be looking in some detail at how those instructions get out of the nucleus and to the ribosomes. And that is a somewhat complicated process, but we'll be looking at that in some detail later on in the semester. Some ribosomes are floating around freely inside the cytoplasm, as these are. Others are basically attached to some ribbony sort of material that we call endoplasmic reticulum. That's what the ER stands for. Endoplasmic reticulum, a great word to sort of drop on your parents sometime if you can work that into a conversation. Really guaranteed to make you sound smart. If you can somehow, work that into a conversation, which, is not easy to do. If you can do that, you probably really are smart. But anyway, so endoplasmic reticulum is the same phospholipid bilayer stuff that the nuclear membrane and the cell membrane are made out of. And as I said, it comes in two flavors. It comes in rough and smooth. The rough endoplasmic reticulum, when it was first discovered, back when we had very, very poor, I think it was in about the 1940s, 1950s, our electron microscopes were not very powerful, and they couldn't really tell why sometimes the ER looked kind of rough and sometimes it looked smooth. Well, as microscopes got better, they found out that the reason it looked rough was because it had these ribosomes stuck all over it. And that's what gave it the rough appearance. And it was still several years later after that that they found out what those ribosomes were actually doing. Okay, so here's our two flavors of endoplasmic reticulum, rough and smooth. The function of the ER is basically to act as a transportation system. Now, I've drawn it as sort of this winding pathway that leads roughly from the nucleus. So if you draw your nucleus here, and then draw the ER as sort of a winding back and forth, sort of a set of switchbacks leading roughly towards the cell membrane. Okay, that roughly describes the path of how the proteins are carried. Remember, the instructions are inside the nucleus. They leave the nucleus, come out to a ribosome, which is often on the endoplasmic reticulum, and then a protein is manufactured along that way. So it's moving, it's actually moving from the nucleus where it's made outward towards the outer part of the cell membrane. Okay, so you can think of the ER as being a little bit like a conveyor belt, and then the ribosomes are sort of like machines that travel along on that conveyor belt and make proteins. Okay, so as I just said, the rough ER is covered with ribosomes. The smooth ER does not have ribosomes. Okay. When we get to the end of our endoplasmic reticulum conveyor belt, we have these little squashed circles, kind of oval structures that we call Golgi bodies or Golgi apparatus, or just Golgis often they're called. And these are, again, structures made out of that same phospholipid bilayer material. What Golgis are involved with is the final stages in producing a protein. As we'll see in a couple of weeks, a protein, first of all, consists of a chain of amino acids, and then that chain needs to get folded into a particular shape. And where that folding takes place, or one of the things that happens to that protein, happens inside of these Golgi bodies. So they're often found somewhere between the ER and the cell membrane. Okay, and that's where I've drawn them here. So again, kind of the image to think of in your mind is a conveyor belt leading from the nucleus out to the cell membrane, and just before that conveyor belt gets to the cell membrane, you have these Golgi bodies that do the final steps of putting those proteins together. Okay, so as I just said, the function of the Golgi bodies is to complete this protein synthesis, complete the process of making proteins and package them for export out of the cell. The Golgi body is named for this gentleman that you see on the right side of your screen. This is Mr. Camillo Golgi. He was an Italian gentleman who lived and worked right between the 19th and 20th century, and as you can see from this picture, he had just a fabulous mustache. He's often remembered for his mustache and sometimes remembered for these things that were named after him. They're probably more famous than the mustache, actually, but, you know, there's just no justice sometimes. Okay, anyway, that's enough of Camillo, and we're done with our Golgi bodies, and I think that's a good place to hang this up for a little bit. I'm going to break this vodcast into two parts. You have just finished listening, watching, and taking notes on VodCast 14A, and now the next thing to look at is VodCast 14B, and I will finish up these animal cell organelles in there. Okay,